Okay. And so go back when anyway, so Mrs. Scott did many, many amazing things in life. And again, remained strongly interested in all things science and loved to talk about, especially genetics. And so I know she is probably watching from heaven and enjoying this talk that we're about to hear today from Karen Mohlke, who is a professor and the associate chair of genetics. She is joining us today from the University of North Carolina at Chapel Hill. Dr. Mohlke got her PhD in human genetics from the University of Michigan, and then she was a postdoctoral fellow at the National Human Genome Research Institute, where she worked with Francis Collins. She joined UNC in 2004, is that right? And has been there almost looking at 20 years and risen through the ranks and is a professor. She's received a number of awards. She was a Burroughs Wellcome Fund Career Award in the biomedical sciences. I did some work for Burroughs Wellcome over the years, so I know that's such an amazing award to have received. A Pew Scholar. She was a Kavli Fellow and is a Fellow of the American Association for the Advancement of Science. She also is now an Oliver Smithies Investigator at the University of North Carolina School of Medicine. So I know that's a great big honor to have. So I'm truly delighted that Karen is here today. We had a great dinner last night, and hopefully all of you will enjoy her talk today, where she's going to tell us about genes and variants that influence common metabolic diseases.
Well, thank you very much for the honor of being invited to present this lecture today. And I've had a wonderful day, enjoyed meeting many people and talking about great science. So I'm going to talk today about common metabolic diseases, type 2 diabetes, obesity, dyslipidemia, related traits, and the genetic approaches that we take to study them. So my group is both a computational side and a wet bench side, and we merge those actions together to learn about the basis of these diseases. So there are many different tissues, many different processes that are involved in metabolic dysregulation. So some of those tissues are shown here, adipose tissue and liver. I'm going to talk a bit about each of those today, as well as skeletal muscle. And there are many processes that are known, insulin resistance, there are many pathways that are known, but we don't know what all of the players are. We don't know what all of those factors are and how they work together. So we take a genetic approach to understanding metabolic dysregulation to learn to fill in those gaps and learn what those factors are. So by a genetic approach, we'll start with, we look at genetic variants, such as this C to T variant in individuals shown here, and measure different phenotypes in individuals. So for example, if some of these individuals had type 2 diabetes and other individuals didn't, we're looking for, does that T allele show up more often in one group than the other? And if that's a significant enough difference, then we say, that variant or one of them that's nearby is influencing risk for type 2 diabetes. So we do this for a range of metabolic traits and diseases. So this strategy is a genome-wide association study. These have been around for about 15 or so years. And I will talk some today about, I'm going to use type 2 diabetes as my example of what we've learned in the process along the way of doing those experiments. However, the genome-wide association studies give us locations in the genome, but they don't tell us what the genes are, what the variants are. So those are some of the big questions that come next. Which genes are responsible for these associations? And which variants affect gene function? And the value of understanding the variants is to have a testable hypothesis to connect the risk to the variant to the gene to understand the basis of disease. So I'm also going to talk about inserting these, figuring out this connection between the variants to the genes. A lot of the variants that we identify in genome-wide association studies are not in the protein coding parts of the genes. And so we think a lot of them have regulatory effects on tuning up and tuning down, how much gene product is present, and their effects on metabolic traits and diseases. So the second part of that I'm going to talk about is integrating this genetic data with gene regulation and gene expression data to be able to understand how the genetic risk functions. Okay, so I'm going to start with type 2 diabetes and talk through genetic association studies in type 2 diabetes. So originally there were candidate gene studies. I think maybe this one might play a role in type 2 diabetes. Oh, there's a variant in this gene. Does that have its effect? The first genome-wide association studies were published in 2007, a series of them. And so here is one of the very first. These are the results from the stage 1 of that study, hundreds of cases and hundreds of controls. And then the chromosomes here are shown separately. And you can see that the many variants are tested on, say, chromosome 1, tested for association, and the red ones were the ones that sort of had the most evidence of association. This y-axis, the strongest evidence of association high up on the y-axis. And the strongest signal in this study was a variant over here on chromosome 10 that was located near the gene TCF7L2. Many genome-wide association studies came out in 2007. Here's another one from the Wellcome Trust Case Control Consortium, about 2,000 type 2 diabetes cases, around 3,000 controls. So I'm going to show a series of these Manhattan plots, and this is sort of the beginning, that there were a signal here, maybe a signal over there. One step that really advanced the growth of doing genome-wide association studies was the ability to test variants that were not directly analyzed in the lab. And so the methods of imputing or predicting the nearby variants had a huge impact on genome-wide association studies. So here, if I have genotypes that I observe, say from a genotyping array at 3 different positions, and I have a reference population of haplotypes much more densely sequenced or genotyped, and then can use imputation methods to identify and predict what the variants are at the other sites. So this is helpful both for increasing the number of variants that can be analyzed and also for letting groups combine their data together if they tested different actual variants in their assays. So genome-wide came to mean more and more as the years went by. So 500,000 variants could be analyzed in those first direct genotyping assays. And then as soon as we could impute, it's imputing to better and better reference panels. So genome-wide association studies were identifying more, both from testing more variants and also as the recognition that sample size matters, and sample sizes were increasing and data being combined. So jump to 2018, and here's a type 2 diabetes genome-wide association study, 74,000 cases, more than 800,000 controls, many more variants tested, and a lot more signals, evidence of association across the genome. You'll see here, it says 403 signals at 243 loci. So I want to explain some terminology because I'm going to use this throughout a lot of the rest of the talk. So here's zooming in what it looks like in one region, type 2 diabetes association. Now we're only looking at 2 megabases of the genome as opposed to 3,000. And the genes now are enough resolution to see below. You can see the different genes that are present. Still, the y-axis is the strength of association for that association analysis. So here we see a region, that top most strongly significant variant. I'm going to call that the lead variant. It's colored in purple. It's inherited together with other variants in the region. Those are colored by how often they're inherited together. So in the beginning, when looking at genome-wide association study data, we'd say, okay, we're going to take that lead variant, define a region around it, maybe 500 kilobases upstream and downstream, say that's one locus, and probably all the variants in there are having the same effect, so we'll just count that and we'll move on. As the studies have grown, sample sizes have increased, we're finding more and more evidence of association. You'll see in this region, there's another variant, other sets of variants over there that show strong evidence of association. They're not inherited in the same pattern as the first set. There's even a strong recombination event that's happening in here. So that's a distinct association signal. So I would say this is one locus, but there's two signals. And those two signals provide an opportunity, more opportunities to learn about the basis of type 2 diabetes. So we can identify those multiple signals based on doing a conditional analysis. I'm going to test for association, but I'm going to account for one association signal to see if there are others in this region. So what's the value of this? Like those signals could be having effects on different genes. So in this case, that first variant, that's right over the GLP-1 receptor. That's a target of a lot of those popular weight loss drugs right now. This other signal, it could be acting on that same gene, or it could be, there's a couple attractive potassium channels right here that could be playing a role in the basis of disease. So by learning about the different signals, we have more opportunities to learn about biology underlying the disease. So that's a little more context for the 403 signals at those 243 loci. So often if there's one variant that could be contributing to an effect on disease risk in a region, maybe there's others that are independent from it that are also having effects on risk. Another advance was looking across multiple populations. So this is data from a long time ago, and it's looking at the ABO blood group and a common allele, the allele that contributes to the A. And you can see the frequency across different populations in the world. And the frequency changes as sort of maybe has changed with migration, the patterns, the evidence that it's contributing to the presence of blood group. The same thing happens with many, many other common variants. And so by studying different populations, different variants have frequencies that are common enough to be detected as evidence of association in different populations. So around the same time as that previous study was being done that was focused on European ancestry individuals, we did a study that was a combined analysis of individuals from different countries in East Asia and looking at type 2 diabetes evidence of association. So the sample size is kind of similar. There's around 77,000 cases with type 2 diabetes, a little bit more than 300,000 controls. This was, and I think maybe still is, the largest genome-wide association study meta-analysis in East Asia. There were multiple biobanks that contributed together with additional studies. So here we've identified more than 300 signals at 183 loci, and 61 of these loci had not been described before at all, even though the European ancestry study was approximately similar in its sample size and power. And even though some of the Japanese biobank data had already come out and identified dozens of other loci for type 2 diabetes. So there's value in looking across the populations. So I'm going to show first the signals that are shared and then talk about some of the signals that differ between populations. And along the way, I'm going to point out some of the people from the lab. A lot of these studies are large collaborations with many people, and I'm pointing out some of the people from the group that contributed and participated in them. So first, we took the variants that were associated with type 2 diabetes in the East Asian population and compared them to what's the effect size in the European population data. So there's a lot on this graph. So the East Asian effect sizes are on the x-axis, and we focused on those. So all those values are above 0, to the right of 0. And then we compared those to the effect sizes in the Europeans. So that's on the y-axis. So if everything was completely identical across populations, you would expect all of the signals, all the variants to be on the unity line here. And many of them are. Many of those variants, if we look at all of the variants together, the correlation value is there's a lot of similarity present there. You see some variants that are off the line. Sometimes the standard error is extremely large. Maybe that's because the variant is low frequency in one population, and so there's more variation in the estimate of how big of an effect it has. If we only look at the variants that were common and significantly identified in both the European ancestry and the East Asian ancestry populations, then the correlation is even stronger. So many risk variants are shared across populations. They're present across populations, and they're common enough to be identified in the studies. Some of them differ, though. Some of these are far off the line, and we can learn from them. So some variants that are not shared, there's a couple examples of variants that are common, have frequencies above 5% in East Asian populations, but are quite rare in Europeans. PAX4 is one of those. And then there are some that are low frequency in East Asians, but still enough to be able to detect that effect, and rare in Europeans. So I'm going to give an example of one of those where the potential effect on a gene was perhaps more obvious from the start, and that is this zinc finger gene 257. So prior to our study, an analysis of the genomes of individuals had been done and reported, and a genome inversion had been described in this one region. There were 2 breakpoints in the DNA. They're about 415 kilobases apart. And so this region of the genome was inverted in some individuals. So importantly, with this inversion, there's a gene, ZNF257, that crosses one of the breakpoints. So you can see that in the standard chromosome, the ZNF257 is intact, and in the inversion, part of the gene is here and part of the gene is over here and headed in the other direction. So the group that had described this inversion had noted that there was an altered expression of some downstream genes, ZNF257, zinc finger, it's probably acting as a transcription factor. They observed that it changed gene expression in individuals that had inherited this inversion. We showed that variants that correlate with this inversion are associated with type 2 diabetes risk. So that adds a genetic component to a mechanism that has been described and suggests, especially since ZNF257 is being disrupted by this inversion, that that gene is playing a role in the type 2 diabetes risk that's represented by this signal. So an inversion is one potential molecular mechanism underlying risk. Okay, so then back to, so that was 2 populations separately, and now here's the current state. Global Genetic Initiative for Type 2 Diabetes, 611 loci, 1,289 independent signals. This is pre-printed analysis, sort of the current largest analysis. So ever increasing the number of signals that are present. So suggesting there's more than 1,000 potential causal mechanisms underlying risk for type 2 diabetes have been identified to date. So back to what are the genes responsible for these signals and which variants influence gene function. So one way we can learn about the potential basis for common diseases is to compare them to the genetic effects on related traits. So for example, the upper plot shows the latest genome-wide association study for plasma fasting insulin levels. So we can compare the association signals for insulin levels to the association signals for type 2 diabetes and learn like which signals are shared. And that can help elucidate what the biology, which ones of those tissues and pathways could be contributing to disease. The bottom plot is the current state of an ongoing meta-analysis of waist to hip ratio adjusted for body mass index. So this is a trait that's looking at sort of central obesity, and the variants that could be contributing to that may be leading or involved in, say, insulin resistance and related to type 2 diabetes. So we can see that there are different signals, some shared signals, and can learn about the biology in that way. So one trait I want to describe a little bit more is looking at plasma levels of proinsulin. This is the precursor to the insulin molecule. Right? Insulin keeps glucose levels in check. Insulin resistance, meaning that it takes larger and larger amounts of insulin to allow the glucose to be taken up by the peripheral tissues and stored until it's needed. So insulin's originally created as a propeptide, and this is in the endoplasmic reticulum of the pancreatic islet beta cells. And it's processed through going to the Golgi and then into secretory vesicles. And then the propeptide is cleaved. Two different enzymes make these cleavages to make the mature insulin molecule and the spare C-peptide. And both of these are secreted into the outside of the cell, into the plasma. And sometimes proinsulin, unprocessed, is also secreted. So high proinsulin relative to insulin can be an early marker of some distressed beta cells. So if we can identify new loci for plasma proinsulin levels, then that can help reveal genes that may be involved in insulin processing or insulin secretion. So we and others performed a genome-wide association study meta-analysis, around 46,000 individuals, and identified signals at 28 loci. So this is about 3 times the sample size observed, analyzed previously, maybe 4 times, and approximately doubled or tripled the number of loci identified. And notably, about 18 of those were new loci for proinsulin. They'd not been described for proinsulin previously. Now, I would claim that proinsulin is a little closer to the genetic effect, the contribution of the genetic variant, than a disease like type 2 diabetes that has multiple parts, multiple tissues, multiple contributing types of processes. We set out to then compare those 18 new loci to, well, what's been known for other glycemic traits? Can we learn about the biology in that way? So for about half of those loci, the 10 shown here, they also showed evidence of association with type 2 diabetes or plasma glucose levels. And in fact, about half of them, the direction of effect was that higher proinsulin was the alleles associated with higher proinsulin were associated with higher glucose levels or higher risk of type 2 diabetes, and the other half, the direction was opposite. So we can start to tease out how the relationship, what the processes might be underlying that's leading to that effect. It's not super simple. It's not just one cause this way, one cause the other. Some of these are beta cell transcription factors. Some of them are playing more receptor type roles. Some of them are maybe influencing insulin resistance that is then leading to distressed beta cells and influencing the proinsulin levels. Potentially more interesting are the 8 loci that had not yet been observed for type 2 diabetes or glucose levels. Maybe these are, if we can see many loci that are shared with type 2 diabetes, and we're finding them in a sample size 1/10 of that for type 2 diabetes, maybe we're finding other loci that have not yet been identified for diabetes that are also diabetes risk alleles. So here are 8 loci where there's a lot less that is known. One of the first things we noticed is PCSK2. That's one of the enzymes that does the cleavage between the proinsulin molecule to help create the mature insulin molecule. So that biology is well known. Now we show that there are genetic variants that influence how that ability to make that cleavage. So that's sort of confidence in biology. While we were doing this analysis, we noticed this variant. If if you look at this, the variance allele frequency is lower than the others, and its effect size is higher, kind of had to be because we were able to detect it. We said, oh, what is this variant? And while we were doing the study, first let me describe that variant. So here's the association signal, and the lead variant is a missense variant in this gene LAPOR1. This is encodes a transmembrane protein, seems like it's involved in the plasma membrane and maybe the secretory vesicles and maybe recycling of those vesicles. And when we look at the characteristics of the variant, it's a missense variant. It changes the amino acid. That amino acid is conserved across species. That change to the amino acid is predicted to damage the function of the protein. And so this looks like a candidate variant that could be responsible for a gene. And while we were doing this, a paper came out that described the role of this gene. LAPOR1 is also known as inceptor. And deletion of the inceptor gene in mice, either in all cells or in beta cells, led to low glucose and higher insulin secretion. So on the left, here's sort of the normal state, and inceptor is this protein is at the cell membrane surface, and there's normal insulin secretion, normal beta cell proliferation. Whereas when it's deleted, then there's increased insulin secretion and beta cell proliferation. So maybe this is involved in the recycling of vesicles to enable the insulin secretion process to be happening. The data from the human genome-wide associations data has a similar pattern of the common allele is associated with lower glucose and higher proinsulin, similar to the mouse. So potentially this missense variant is influencing this gene to lead to effects on insulin secretion. Okay, so we've seen an inversion and a missense variant that could be playing a role. But a lot of the time, the protein coding sequence is not, none of the variants are located in those regions. So how do we link the variants to genes in those other cases? So we do that by integrating with gene regulation and expression data. So I'm going to take a genetic approach to analyzing gene expression data. So we're going to look at genetic variants, such as this T to C allele, and the expression level of genes across the genome in specific tissues to ask whether one allele leads to higher expression than the other allele. And then asked, do the same genetic variants that are associated with risk of disease, are they also associated with levels of gene expression? And if so, then we can hypothesize that the variants act through those genes potentially to influence the traits. So I'm going to tell one story like that. And then how does the gene expression change? Well, some of that's through the gene regulation process with chromatin. It's a little closer to the effect of the genetic variant. So we're going to look at genetic effects on chromatin that can be leading to effects on gene expression and influencing traits. Okay, so the first study is focused on gene expression. So we're going to analyze gene expression quantitative trait locus, or EQTL, analyses. So just like a genome-wide association study, I'm going to take variants across the genome, and in this case, subcutaneous adipose tissue, analyze with RNA levels measured by RNA sequencing, look at around 30,000 genes or transcripts, and test for association. And since we care most about understanding disease risk and those GWAS signals, we're going to focus on the genetic variants located within about a megabase of given genes to try and figure out how the variants could be having their effect on traits. So we did this in collaboration and did a EQTL meta-analysis, combined 5 data sets together. So Sarah Brotman in the lab, we did this together with colleagues at the University of Michigan and colleagues at King's College in London. So we combined the data from 5 different data sets, And the total sample size from these 5 data sets is around 2,200 samples. And we, with this greater power, because with a larger sample size, we chose to identify and specifically try and find the separate signals that influence expression of each gene. So you could imagine a promoter variant influences expression of a gene, and maybe there's an enhancer that influences expression, or 2 or 3, or maybe there's a repressor that influences expression. So we teased those apart in the identification and meta-analysis. So here's what we saw. First, if we focus on the genes, those are the asterisks, you can see that the number of genes that were identified from each of the individual studies, shown here on the y-axis with the different colors corresponding to the different studies. And you can see that there was an increase in the number of genes identified. And there was an even greater increase in the number of signals that were identified. So overall, we identified 34,000 EQTL signals for more than 18,000 genes in this more powered, larger study of adipose gene expression levels. We characterized those some. The one piece I'm going to show you is we looked at where those variants that influence gene expression, where are they compared to genes? And we found, and we separated them by the order in which they were discovered. So the first signals that are found, these are the ones that are found in a smaller initial study. And we found that we looked at 600, a little more than 600 genes that had all had 5 association signals. And for those 600 genes, the first signals are more often in promoters. They're highly enriched in promoters, and they're also enriched in enhancers. And the second, third, and fourth signals also significantly enriched in both promoters and enhancers. The first signals especially so in promoters. So a lot of the signals that are identified first, those are the ones that maybe even more likely shared across tissues. These are the promoter signals for genes. And the ones identified later, some of these are further away and maybe are more likely to be in enhancers and the regulatory elements that are harder to find. Okay, but what we really care about is linking those gene association signals with the trait association signals. So the strategy we do that is to do that is called co-localization. And the idea is in these plots, if there's an GWAS signal in a region and there's an EQTL signal in a region, I can't just take the GWAS variant, look it up in the EQTL data set, ask if it's significant, and say, aha, I think these are the same. Because the variants that are responsible for this GWAS signal, in this case, don't look like they're likely to be the same as those that are the lead variants more likely to be responsible for the EQTL signal. What I need is those signals to be having the same pattern of association. So we're again asking whether the variants associated with the traits are the same variants associated with the gene expression level, hypothesizing that the gene can affect the trait. So we did this analysis by isolating the distinct signals through the way that we identified them, and then we used conditional analysis so that we could look at the first signal conditioned on all the others, then the second signal conditioned on all the others, the third signal conditioned on all the others. And then we did the same thing with the genome-wide association study data. If summary data was available, if the signals had been identified, we used those. If they weren't, we identified them and then conditioned on all the other signals. So now we can compare isolated signals, in part because the co-localization method works better this way and it gives more accurate results. Then we filtered the signals going in, which ones we were going to trust to evaluate based on the linkage to equilibrium, the inheritance pattern relationships between them, and then used a program called CoLoc to identify the ones that we believe are co-localized. So we did this for 28 traits. 13 of them are shown on this slide. So when looking at genome-wide association studies signals for, say, high-density lipoprotein, HDL cholesterol, we identified 369 pairs of GWAS EQTL signals that were shared or co-localized. Those correspond to the numbers shown here for the numbers of genome-wide association signals. So here, 213 association signals, and they correspond to 344 genes, because sometimes the same genome-wide association signal co-localizes with an EQTL for 2 nearby genes. Totaling up across all the 28 traits, there's 1,800, more than 1,800 genes where the pattern of association for the gene is the same as the pattern for association for the trait. So these 1,800 genes are candidates now for potentially playing a role in these traits or diseases. On average, we found that when we focus on the genome-wide association signals, about a third had at least one co-localized EQTL signal. This is a lot more than when we did this previously with only one of those 5 studies. We about doubled the percentage of GWAS signals that we could identify a gene for, a candidate gene for through this approach. And for the traits that were especially related to subcutaneous adipose tissue, some of these depot traits especially, about 2/3 of the signals had at least one EQTL in this analysis. So we know that the EQTL analysis is still underpowered, but we're making great progress towards identifying potential genes in this tissue that could correspond to these traits and could be playing a role in these traits. Also, because we'd gone through the work of separating out those signals, we said, well, how many did we find because we analyzed the separate signals? And it was almost 50% more co-localizations than if we had just focused on the first signal that was found for, say, each gene. So we're finding a lot of association signals. Can we start getting towards causality a little better? So we used a Mendelian randomization approach called MR-Locus, developed by my UNC colleague, Michael Love. And shown here are 4 positive control examples. These are 4 GWAS loci for which co-localization has suggested a particular gene plays a role, and for which biology experiments that have manipulated the gene have shown evidence in support of that gene. So first, on the left, here is EQTL signals for lipoprotein, LPL, lipoprotein lipase, and the GWAS signals coronary artery disease. So the plots are showing on the x-axis the effect size of the EQTL, and the y-axis is the effect size of the genome-wide, the risk for the trait. And you can see that each dot here, each is representing a signal. So we saw many signals associated with LPL gene expression levels, and many signals associated with coronary artery disease right in this region. And they all have the same direction of effect. And it's a pretty similar effect size relationship. So we're learning a little bit about the gene dosage effect on the trait. So I think this is well established that LPL plays a role in coronary artery disease, but here's an example of a biology that said, what's the effect of altered LPL on risk? And here are 3 other examples where biology experiments have suggested the role of the gene, and the EQTL to GWAS relationship is in support of that. So those are some of the established signals. We also then looked at the genes that we don't know so much about. And so here's one example, and I show just a little bit more. There were 2 association signals for waist-hip ratio in about 500,000 people. They're shown here as one shown in red and one shown in blue. Here's the adipose expression data for the gene ZNRF3. It's located right here. So there appear to be 2 signals that are associated with the expression of that gene that match the signals for central obesity, waist-hip ratio. And this gene has been shown to negatively regulate Wnt signaling activation, and Wnt signaling has been implicated in other loci for waist-hip ratio. And the effect sizes, the dosage effect of the gene corresponding to the trait. It's a wider range than some of those examples I showed you before, but the signals are sort of acting in the same direction. So you could imagine that a drug company that's trying to target a gene would like to know, how much do I have to change the amount of this functioning gene to have what kind of an effect on a phenotype? So we could start to make some estimations here from the genetic data to address those kinds of questions. Okay, and one more thing we did was look at regulatory variants, trying to identify variants that could be responsible for these signals. So here's an example. There's a waist-hip ratio signal shown on the left. Again, about 500,000 individuals. And here's the EQTL data for the gene SEMA3C, same lead variant between the two. And we asked, how might this variant be having its effect? We compared the location of those variants to gene regulatory information. So on this first plot, the variants are shown right here, and they're located a little bit upstream from the gene. And if we zoom in, then we can see that the variants are located in this region where the regulatory evidence is stronger in mature adipocytes than immature adipocytes. And so then we can ask, does one of those variants play a role in changing transcriptional activity to lead to the effect on gene expression? In fact, about 70% of our adipose expression signals had at least one variant that was located in a region of adipose or adipocyte regulatory information. So we tested this particular variant. We cloned the region around, right around that variant, and we cloned it once with the G allele, once with the C allele, upstream of a reporter gene, luciferase, and asked how much transcriptional activity is happening here, and is there a difference between the two alleles? And what we saw is that, yes, this element acts as an enhancer, and that there's a significant difference between the two alleles in how much enhancer activity is present, regardless of the orientation in which we clone that sequence. So we can use this data to learn about the genes that are playing a role, that could be playing a role, and finding particular variants that could be responsible for those effects on genes to help start to build mechanisms. Okay, so through the adipose gene expression data, we found that the larger sample size finds more signals than any of the individual studies, that they're further away from the transcription start sites of genes, so we're learning a little bit more about gene regulation at a distance, and that by comparing those to the genome-wide association studies, we're finding more than 1,800 candidate genes for playing a role in one of these cardiometabolic traits. Looking at those signals separately helped us in this discovery. And when we look at the pairs of variants, when there's two or more signals, and we look at those dosage effects of the genes on the traits, we can learn potentially about more causal directions. So lastly, I want to say, okay, so we looked at gene expression. Now let's look at gene regulation. And I'm going to tell one story using liver genomic data and looking at gene regulation to influence gene expression. So that's this chromatin context. And again, we're looking at genetic variants that are associated with the chromatin and how the chromatin is leading to gene expression. And the assay that we use is this assay for transposase accessible chromatin. So normally the DNA is wound tightly around histones, but then in some places it's open, it's accessible to like this blue transcription factor's ability to bind. So the transposase can come in, release these open accessible chromatin regions. We can identify them, sequence them, map them back to the genome, and identify regions where they build up those that are representing, we think, regulatory elements. So we did this for 138 individuals. We previously did it for a sample size of 20, and that's published. The 138 is still ongoing analysis. In those individuals, we find around 350,000 of these regions, potential regulatory regions in the liver. So to look at genetic variants that influence these regulatory regions, the analysis I'm going to focus on is when the variant is located in the region or very close to it, within 1 kilobase of that candidate regulatory element. So in our 138 individuals, we found 35,000 regions across the genome where a variant is in or located quite near a regulatory element that shows differences in accessibility. Here are the profiles across one region from 9 different individuals. So the places where we see these peaks are where those reads build up. These are the regulatory elements. So in some places, the 9 individuals, pretty similar pattern across the different individuals. Whereas in some regions, like the one over here, some individuals have a larger peak, they have more accessible chromatin than other individuals. These genotypes over here are the genotypes of a genetic variant located right in this purple region right here. So individuals with the T allele are more often have accessible chromatin, individuals with the C allele less often accessible chromatin. This is what we're looking for. This is our evidence of association between a variant and a trait, but in more individuals. So maybe the variants are revealing the location of regulatory elements that they're directly related to. So our association analysis, we look at the chromatin accessibility QTL, and then compare those to our genome-wide association study signals. So can we identify variants in regulatory elements at the GWAS signals to learn how they could work? Same sort of approach as before. We're looking for co-localization of these patterns of signals. So here we focused on traits a little more related to the liver than to the adipose. So here are the series of genome-wide association study traits we analyzed. And shown here now are the number of genome-wide association signals that we looked at, the number that had a co-localized chromatin accessibility QTL. And for the traits more related to the liver, we see higher percentage of signals that show co-localization. What this really means is we have potential candidate variants that are playing a role in a regulatory role at these genome-wide association signals. So we're getting the regulatory the variants, potentially the regulatory elements, and maybe pinpointing a little faster to the potential mechanism underlying how the gene regulation could be influencing the trait. So I have one example I'm going to show with alanine aminotransferase levels. So in this case, the GWAS signal shown in the upper left in hundreds of thousands of individuals has the same pattern as a liver expression quantitative trait locus effects for the gene EFHD1 in around 1,100 individuals, similar pattern of association. Here's the chromatin QTL data. This is actually from the 20 individual sample, 20 individuals, and you see the same set of variants show the strongest evidence of association. So the variants that are influencing chromatin accessibility, including the variant in the regulatory element that's right here, is the strongest evidence of association with the trait levels and one of the strongest associations with gene expression levels. So we hypothesize that that is the variant that's influencing gene expression to have an effect on the trait. So we did some experiments like the ones I showed before. Yes, that variant can influence transcriptional activity. Yes, it binds better to one transcription factor, FOXA2, than another. And then we wanted to say, but can we link the regulatory elements to this gene? So in this case, we used CRISPR interference, and we used guide RNAs located at the location of this regulatory element, and use those to recruit a repressor. We used a dead Cas9 with a crab domain repressor to this region and asked what the effect was on expression levels of the genes nearby. So the guide RNAs are shown here, sort of spanning that regulatory element. We had cell lines stably expressing this Cas9 with a repressor domain, and then provide these guide RNAs compared to guide RNAs that don't target any regulatory element in particular, and asked, what's the effect on gene expression? And we're going to ask it on this gene and the two nearby genes. So the gene that's closest here, it looks like knockdown of this regulatory element has about a 20% reduction of gene expression of this gene. So how much is the promoter influencing? How much do different enhancers influence? But this enhancer, from this experiment, we would suggest can influence gene expression of this gene. Whereas the two nearby genes, we don't see significant differences in the effects on those genes. Now, sometimes regulatory elements do act on multiple genes, but this is one sort of data that helps build the story of the mechanisms of how these variants can function. So in this case, we propose then from these data that this particular variant that's located in a regulatory element that showed the T allele showed more chromatin accessibility. This is also the allele that bound to the transcription factor FOXA2 better, that also showed higher transcriptional activity, that also in the human liver data shows higher expression of the EFHD1 gene, that also corresponds to higher levels of alanine aminotransferase. So then we can propose a mechanism that this variant could be responsible for this association signal acting through this gene. So with this gene regulation data, DNA variants can have a strong effect on chromatin accessibility, potentially by binding these transcription factors, stabilizing the chromatin in a state that's accessible. And we found tens of thousands of CAQTL in a set of individuals, and that these are strong candidate variants for effects that could be contributing to the genome-wide association studies. So we can combine data from the CAQTL, from transcription factors, target gene predictions to identify potential mechanisms that can then be tested in the lab. And then, like the previous study, moving towards separating out the cell types within the human tissues can help us understand which cell types within the tissues are contributing. So overall, we can use genetic approaches related to traits and identify hundreds of loci for each of the many metabolic traits, use the gene expression data to identify candidate target genes in some of the relevant tissues, use the gene regulation data to help build potential mechanisms by which these act, hopefully identifying then good targets that could be used for drug discovery, understand better the biology of the genes that play a role to hopefully reduce the risk and impact of disease. I've shown you a lot of studies with a lot of collaborators, some of which are on the slide here. Some individuals from my group, present and former, that contributed are shown here, and then some of our collaborators with the various studies that I spelled out the most. So thank you very much for your attention. Thank you.
We're open for questions. I'll start a little. I mean, I love the studies on human tissues. I'm going to guess your adipose tissues were from normal people or from diabetic people. And how do you think it would change if it were one or the other? Or can you separate those and ask the questions?
Right. So both are interesting. Our studies are largely from individuals who are from population-based studies. So and in like the metabolic syndrome in men studies, we specifically avoided people with disease because we didn't want to create complications. So they could walk into the clinic, provide the tissue sample, walk out. That provides a context of the tissue that may differ from, say, tissue collected from individuals that, say, are extremely obese and undergoing gastric bypass surgery and have tissue collected. Now maybe the inflammatory environment is different, the transcription factors are different. We're going to find different signals. Both are interesting. I would say the one without the disease setting maybe helps you get more easily at the true genetic effects, but without the contamination of the different variation in the biology that's happening. But the disease state is needed because that's how some of the variants are going to be functioning, is because of those effects. Yeah.
I was thinking the disease state might actually get you to disease state relevance more, right?
Right.
Yeah.
Right.
And then other questions while you're getting, oh, there we go. Go ahead, please.
Measures factors, curious about like eight-factor analysis about curious about the power
Summarize the question.
Yeah, I think you're asking, a lot of the traits are related to each other. And so can we use various factors that are contributing to the combination of traits and use that as a strategy to look for, say, the genetic effects on those factors? So yes, the practicalities of collecting the data at the genome-wide association study stage are that often there's like 200 studies contributing data. And so it's a process of asking everybody to follow that analysis plan and collecting that. So the stuff that's quicker is the more straightforward analysis, and the stuff that, like, that analysis would be provide instructions to those analysts to generate that data to combine that. So less has happened in that direction, but it's a way to learn, especially about certain angles of which, right, especially with heterogeneous diseases, to sort of tease apart those contributions.
Beautiful talk, thanks. Can you say something a little bit about diverse genomes and like where you see this going? It's very impressive, the novel loci by going outside Europeans, but maybe just a little perspective as to what you see coming down the pike here in this area.
Right. So genetic variants, right, across humans, there's a whole range of variants, and just because one population has a higher frequency of some common variant, like the biology that's uncovered is relevant to all. There are variants that we're able to identify, like they need to be common enough to identify them in the sample size that's available. So the diversity, the potential for the number of variants that we can learn about in genetic approaches is enormous. Probably every variant that is compatible with human life is present in an individual alive today. Can we get enough copies of those in a population to be able to study and look at their effects? Right, we're doing these natural genetic studies, making use of those. So it's huge, and there's so much more variation present, especially as we're getting into the lower frequency variants that can be identified in populations, that I think the potential for biology that we can learn is just going to greatly increase.
Yeah. So actually similar to our question. You showed a which trait variants with different effect sizes in two different ancestries, right? Do you think that's actually do you think the underlying biology is actually the same, and that's just another way to discover it, and it might be shared in the population, and maybe you just don't get the signal because it's washed out and not enough carriers or something in one ancestry? Or do you think it's like, you see what I'm saying? Do you think it's biology specific to that population?
Right. So the question, right? A lot of the effect sizes were shared. Some of them looked different. So some of those, the error bar was huge in the other population. And so it's not a significant difference yet. But what could be the cause? There are variants that are off the line and have narrow error bars in the different populations. So one possibility is that there's a nearby variant that's influencing this one, that's influencing the trait that we don't know about, that we haven't been able to account for. And so the effect size that we're measuring is not really the effect size of that variant. It has some contribution from another variant nearby. we did look at some cases like that and try to figure out, is there another signal there? I'm like, maybe. And we don't have strong evidence yet for any of those as sort of playing a role, but those are probably out there, right? It's hard really to separate the signals properly and evaluate those. The other is there some sort of shared environment, or is there some other locus where maybe like a transcription factor that there's a higher expression, a higher presence of that's more common in one population than the other, and that's playing a role influencing the effect size. So you could call that environment, or maybe it's genetics leading to environment that is contributing to those effects. And I think that's possible too, right? It has to be a common enough factor that you're seeing it when you look at that population to be having a role that way. But I think both are possible, and probably the combination of those is possible. And underlying, I think the variants are doing the same thing, but maybe there's settings that are influencing how much that's happening in a given, study.
All right. I don't see questions online. Okay. Well, let's, so first of all, I think Kiara is outside. Kiara is the person who arranges this whole day. Oh, there she is back there. Thank you, Kiara, for all your work. And thank you, Yesenia, for all your help as well. And then let's thank Karen for a great talk. And please join us for a reception outside where you can ask Karen lots more questions. Or not, just enjoy the food. Oh, a small gift. And a gift for your troubles. Who doesn't like a blue bag?